molecular phenotypes to understand underlying endotypes and how we can start targeting unique mechanisms for treatment. So, for those of you who don't know, so inflammatory bowel disease encompasses both ulcerative colitis and Crohn's disease. I'm going to talk about ulcerative colitis today. Brief background: ulcerative colitis, immune-mediated inflammatory disease of the colon, mediated through background genetics, microbial host interactions, and immune processes. What's really relevant here is we don't have a single process that drives the disease inflammation. We have a multitude of observations that have been made for why ulcerative colitis inflammation is occurring. Accordingly, we have multiple therapeutic options that have been developed, and IBD is one of the fastest growing pharmaceutical development spaces with billions of dollars of investments and probably over 24 therapies now available. However, every single therapy that we have, despite the fact that we have over 10 different mechanisms of action that we use, only work about a third of the time. So every time we use them at a population level, it's about a third response rate. And so that results in a lot of trial and error approach for treatment. And despite our best efforts, we've been unable to predict treatment response using baseline inflammation. And we also have had tremendous difficulty in explaining treatment failures. So those patients where we would have expected them to do well, they don't do well, and we don't have a clear understanding, and that's been a combination of our lack of understanding for the disease and a lack of technological innovation for how we study the disease. And so our remission rates are abysmal. We're looking at maybe 20, 30% over 1 year, right? Compared to a lot of other immune-mediated diseases. One of the core problems for inflammatory bowel disease is that we still rely heavily on a phenotypic classification of the disease, and we use a phenotypic manifestation of the inflammation to define treatment response, often at a single time point, right? And if you think about that conceptually, patients heal at different stages, wounds heal at different stages, they take different time periods to heal, they heal through different mechanisms. So if you apply a universal one-size-fits-all phenotype and a universal one-size-fits-all time point, you're not going to capture the granularity in that heterogeneity of response trajectories. And it's currently our endpoints are drug agnostic, which means we apply them for every mechanism regardless of how they work. So what we started doing is we started actually partnering with a lot of the pharmaceutical companies to be able to gain access to the phase 3 clinical trial data sets with our international collaborations through the 3TR consortium in Europe, and try to build a refined approach for how we want to discover mechanisms of treatment resistance through trajectory modeling, right? So really latent space modeling, trajectory modeling, and start to develop drug-specific models where we can model the interaction between the patient and the therapy and link it to the underlying understanding of the disease process. So that's what I'm going to talk to you about today. This is all unpublished work, soon to be hopefully published in the next month or so, but just to give you a framework for how we're thinking about reinventing the process. So to orient you on what we used, so we gained access to 2 large phase 3 registration trials for vedolizumab. So what is vedolizumab? It's an anti-alpha-4 beta-7 integrin. It targets the alpha-4 beta-7 MADCAB interaction that's specific to the gut. We took a head-to-head comparative trial of vedolizumab versus adalimumab. Adalimumab is an anti-TNF therapy, so anti-cytokine versus anti-trafficking, in a randomized head-to-head trial. So both arms of that trial are now balanced for both measurable and unmeasurable confounders, right? And then we had a second validation cohort with the original placebo-controlled trial for vedolizumab called Gemini, where we were able to take our observations and confirm them and validate them in a second trial data set. What was unique here is we got access to the patient-level data, but we also got access to all the molecular data for bulk RNA sequencing. We got all the O-link data that was generated from it, and then we were able to link it to real-world data that we had in the lab, as well as published single-cell and spatial data, so we could start to think about not only what are the underlying modules of inflammation promoting resistance, but what's the cellular source? How is that cellular source interacting in the spatial environment to promote that inflammation? And at the end, I'll talk about how we've identified a way to overcome that through a repurposing of a commonly available medication. So these are the 4 reproducible vedolizumab response trajectories. How do we do this? We use group-based trajectory modeling, modeled on patient symptoms. So really simple: rectal bleeding, how much blood are you noticing in your stool, and stool frequency. So 2 regulatory accepted endpoints. And what we did is we allowed the patients to cluster out into subgroups, and we found in the first data set I'm going to try using the pointer. Did it work? Yep. So in cohort 1, which is the varsity head-to-head trial, you can see these 4 trajectories really separate, but I'll call to your attention 2 things. 1 is the timing of the separation, and 2 is the slope of separation, okay? You see this delayed slope of separation and a smooth slope for this anti-trafficking therapy. When you go to cohort 2 with Gemini, you can see something similar in terms of the separation, the proportions of patients. You can see this separation, the slope. And for an anti-trafficking therapy, we have 2 very clearly trajectory sort of driven components, validated in 2 trial data sets that were prospectively collected with routine patient-reported outcomes. When we then looked at adalimumab, the anti-cytokine therapy, you can see here when we modeled out the trajectory of these patients specifically in that trial, the trajectory slope was very different. You see very rapid separation between the patient trajectories. You see the slope trajectory is quite unique for each of the different slopes, and you can see that the separation among the 4 groups within the adalimumab group was quite different than what we saw for the vedolizumab group from the same trial. So again, remember, it's a randomized head-to-head trial, so it's balanced for measurable and unmeasurable confounders. And so this began to demonstrate for us that even simple patient-reported outcome dynamics can help separate patients on their response trajectory based on the overall slope and the trajectory that they're going on. And this is class specific in terms of what those dynamics look like. The next thing we needed to do was we needed to link these trajectory response models and these early patient-reported outcome-based trajectories to long-term disease outcomes. So we were able to access both the molecular and sort of phenotypic data, but also the long-term safety data. So on the left here, what you're going to see is we looked at the different trajectory models for each of the drugs. So V1 through V4, the non-responders, and A1 through A4, V being vedolizumab, A being adalimumab. And disease clearance here is actually full, complete disease remission. So symptoms are gone, endoscopy is normal, histology is normal, quality of life has returned to normal. And you can see a very nice relationship or association between the trajectory groups as they were defined by the early symptom changes and the 1-year outcomes for disease clearance. Then when we go out to the Gemini data set, which was the validation data set, what you can see here is that we can actually link these early patient-reported outcome changes to long-term risk for hospitalization and colectomy. And you can see that these early patient-reported symptom changes do link to long-term disease complications. So the first step was, do we have drug-specific separation in trajectory modeling, which we do. The second step was, do these drug-specific changes in trajectory modeling link to overall long-term disease complications while on therapy? That was a big part of it, is we wanted to make sure we understood mechanisms of treatment failure, and so we wanted to link it to actual development of complications despite the use of active therapy. And that's where then the fun became, is we started to then want to understand what are the mechanisms of resistance that we can